Hello, everyone. We're going to wait for maybe a minute so that people can join, and then we will proceed with the webinar. Okay, so I guess we can proceed now. Hello, everyone. My name is Camelia, and I will be the host. Today, our topic is an update on primordial dwarfism conditions. All questions can be asked in the question box in the upper right corner. I want to say thank you to MTG5 for organizing this webinar. Our chairs today and MTG5 representatives are Susan O'Connell and Charlotte Hoiby. So without further ado, I will give the word to Susan O'Connell to present our speaker, Andrew Jackson.
Thank you, Cami. Welcome everybody on behalf of Endo-ERN and our working group of growth and genetic obesity syndromes. We're delighted to welcome Professor Andrew Jackson from the University of Edinburgh. He's a program leader at the MRC Human Genetics Center, and he has a background of clinical and research postgraduate training in Newcastle, Leeds and Sheffield, with his PhD undertaken in the laboratory with Professor Jeff Woods on the molecular basis of primary microcephaly. Over the past 15 years, Andrew's research has focused on the identification of genes for inherited neurological disorders and in defining the functional role of the proteins they encode. He has also discovered over 20 human diseases acting in growth and inflammation, all involved in fundamental cellular processes. And from the starting point of human disease, his research goal is to provide new insights into basic biological processes. And we're very excited to welcome Professor Andrew Jackson to talk to us today about primordial dwarfism and how this impacts on our patients. Thank you, Andrew. Over to you.
Well, thank you very much for this invitation to speak to you all from Charlotte and from Susan. I'm a clinical geneticist and practice here in Edinburgh. And what I'm going to talk to you about today is essentially one half of my lab's work for the last 15, 20 years, which has been on primordial dwarfism or microcephalic dwarfism conditions. So before I get started, just to say that I'm going to show you a lot of patient photography today, which for some of them I have consent for presentation only, so please don't capture or save patient images from this talk. So to start, so the group of patients I study are, in media speak, the smallest people in the world. This lady here is very well known to me from patients' support group meetings. She's an adult in her 30s. She drives her own car. She volunteers in an elderly daycare facility. And I've had lots of lovely conversations with her. And she doesn't look particularly remarkable until you stand her next to an age-matched control. And Louise here very kindly has volunteered to be the age-matched control. And Louise was a postdoc in my lab who discovered several of the genes I'm going to talk about today. So as you can see, it's as if this other lady has been shrunk in a three-dimensional photocopier, and she's in proportion, which is unlike other forms of dwarfism. So this is a scaling disorder, and it's a scaling disorder due to reductions in cell number. So this is a group of conditions which are hypocellular dwarfism disorders. These conditions have a genesis very early in human development, such that by birth there is severely restricted growth. So these children are born small for gestational age, as you can see here from this scale bar of a biro pen. The other thing which distinguishes them from other forms of dwarfism, as well as their proportionality, is a reduced head size. So as you can see here, this lady here is stood next to her friend who has achondroplasia. And you can see a significant distinguishing feature here is head size. This is something which isn't really readily apparent to us when we look at people, because we're used to making adjustments in people's body proportions in terms of head size, in that children are small, are large-headed and a small body, and adults, normal adults, are small-headed on a big body. So this brings me to a major diagnostic point for these conditions, is that you will not make this diagnosis unless you measure head size. And this is important to measure even in individuals who are over 2 years old or in adults to make this diagnosis. And this distinguishes microcephalic primordial dwarfism from conditions such as Russell-Silver syndrome or 3M. Here, I will describe conditions where typically head size is in proportion to body size, or in some cases, head size or microcephaly, as measured by occipital frontal circumference, is reduced relative to body size. The other thing to note here, as in the picture I illustrated earlier, is these are disorders of extreme growth reduction. So if we think about these conditions, we're defining operationally these individuals as over 4 standard deviations, or a Z-score of -4 from the population mean. This group of disorders, my personal excuse for studying them and for in funding bids is to think of these as a human as a model organism concept, in which we could understand factors which determine body size, which is an important question in mammalian biology, which remains unresolved. We're very much aware within species of differences in size, but in mammals, the greatest difference between mammals is that of size, with a 7 magnitude difference between the smallest mammal, the bumblebee bat, at 2 grams, and the largest 150 metric tons. I will come back to this towards the end of the talk, but my brief for this presentation was to the clinical insights we've got along the way. So as Susan said at the beginning, we've discovered a lot of genes along the way for this group of conditions. Maybe not the genes I expected to find, but they have informed us into the mechanisms determining size. So let's start with thinking clinically about diagnosis of these conditions. So that there are a large number of genes from monogenic disorder means that this is actually a group of conditions where mutations in any of a number of genes cause this phenotype. There are forms of this condition which can be recognized clinically as recognizable syndromes, such as MOPD2 or microcephalic osteodysplastic primordial dwarfism type 2, MOPD type 1, also known as Taibi-Lindau syndrome and Meyer-Gorlin syndrome. And I'll go through the phenotypes of these conditions as we go through this talk. There are also, and probably the predominant number of diagnoses, are molecular diagnoses rather on the clinical phenotypes. And these we often describe as microcephalic primordial dwarfism not otherwise specified, or link microcephalic dwarfism with the gene in question. There are also other disorders which overlap this group of conditions in terms of size, which include syndromes such as Bloom syndrome, Cohen syndrome, or Cornelia de Lange syndrome. Typically, these disorders are autosomal recessive in inheritance. There are a few examples of X-linked conditions, and also of de novo dominance, which I'll come to later in the talk. So if we think of the last 20 years, there's been a substantial number of genes identified by us and others, and using our cohort, which is now of over 800 families collected worldwide from clinical geneticists, endocrinologists, and other clinicians who have been generous in sharing their cases with us, we now have about a diagnosis in about half of cases. We can think of these in terms of the syndromic diagnoses I've discussed, and also themes in terms of functions of what the genes do that cause disorders. And as well as molecular diagnoses, it's important to also point out that sometimes we make a diagnosis on the basis of a microdeletion, so a chromosome abnormality, and I guess the one which sticks at my forefront of my mind is a 4p minus has phenocopied primordial dwarfism on several occasions for us. So the other way of thinking these conditions is in terms of the molecular diagnoses. So for instance, MOPD2 is due to mutations in a gene called pericentrin. There's a group of replication genes which cause Mayer-Gorlin syndrome. The non-coding RNA, RNU4 ATAC, causes MOPD type 1. And then there's a whole range of other molecular diagnoses which have been made with relatively common genes involved in a process like centrosomes, non-homologous end joining, active replosome genes, etc. So to help sort of bring this together rather than giving you a whole list of 40 to 50 genes, let's think of this in terms of underlying mechanisms at the cell level. So firstly, there's a group of genes such as pericentrin, PLK4 and TUBGCP6, which are involved in the process of cell division, mitosis, the stage where chromosomes are segregated equally into daughter cells. As well as these, the other theme which comes along is DNA replication. Both the processes in G1 of the cell cycle, where replication licensing occurs, where origins of replication are defined by the ORC proteins CDC6 and CDT1, and then the assembly of this machine called this CMG helicase, which unwinds the DNA such that the polymerases pol alpha, pol delta, and pol epsilon can then copy the DNA to achieve two accurate copies of the DNA that then are contributed to daughter cells. The third theme which has emerged from the genes we've identified have been genome stability genes. These have included genes which are involved in joining DNA which has been broken apart, non-homologous end joining, such as ligase 4 gene and XRCC4, as well as genes which are involved in ongoing DNA replication and also the unraveling of tangles in the genes, such as topoisomerase 3. Finally, there are genes which are actually also fundamentally cell cycle regulating genes, such as CDK4, which we described this year as a cause of microcephalic dwarfism. So what do all these genes have in common? Well, essentially, they all appear to act at different stages in the cell cycle, and when mutated, they will impair the progression of cell cycle such that cell proliferation, the generation of daughter cells, is reduced in efficiency. This leads us to a simple model in which these conditions, which are due to reduced cell number, are the result of slowed cell divisions during development. And with less cell divisions, you end up with a smaller person. With less cells generated, fewer cells generated, you're unable to build a person of normal stature. So this links evolutionary in that there's scaling in mammals, for instance, between man and mouse. The 2,000-fold difference in size between mouse and man is reflected by a 2,000-fold difference in cell number. Likewise, this model also explains for microcephaly in that these genes are acting to affect cell proliferation right from conception onwards. And there's this critical period of neurogenesis in early fetal life when essentially all the neurons in your brain are generated, where these genes will be active and reduce head size and body size at the same time. Whereas genes acting later in development and post-birth, for instance, in the context of growth hormone, will not affect head size in the same way. Okay, so that's an overview of diagnosis without going into the detail of all of the genes which have now been identified for this group of conditions. So what about management? Well, I think there's a couple of general principles to touch on here. The first is, because these are hypercellular disorders, increasing nutrition early in life, unlike in other disorders, does not result in increased growth, nor typically does growth hormone treatment affect final stature. And this is due to the failure of cell proliferation, with fewer cells leading to a small person not being compensatable by increasing nutritional input or growth hormone stimulation. There are a couple of exceptions to this, where there are mutations in the growth hormone IGF-1 axis which are amenable to therapy. One of the places which, one of the genes which, when mutated, can cause microcephalic dwarfism is mutations in IGF-1, where biallelic loss of function mutations cause microcephalic dwarfism with a severe mental retardation and sensory neural hearing loss. The other general management point is that there are patient support groups both in the UK, which also has a lot of European patients in it, the Walking with Giants Foundation, and also in the US and wider afield, there's a Potentials Foundation, which is a grouping within the Little People of America. So now turning to the specific syndromic forms of primordial dwarfism and specific management points. And I think generally management is informed by molecular diagnosis, as I will illustrate in the next few slides. So the most common form of primordial dwarfism is MOPD type 2, due to mutations in the pericentrin gene. So pericentrin encodes a protein which is present in the centrosomes. These are the regions of the cell which focus the mitotic spindle and ensure efficient cell division. As you can see here, facially, there is a distinctive facial appearance which is apparent across different ethnicities. Here you can also see this phenotype in the same individual across ages. It can be recognized also by other features such as very small, very loose dentition, which is present, including in secondary dentition, as you can see here, and also these brachymesophalanges, these very short fingers and slightly wrinkled skin appearance in the hands. Along with this, there are significant clinical complications. The most concerning for patients and their families are the vascular. So from childhood onwards, there is a significant risk of neurovascular disease with cerebral aneurysms and also moya, causing a significant neurodisability. As well, in early adulthood, chronic renal disease becomes an issue, as does ischemic heart disease. And lifespan in many of these individuals does not progress beyond 20s, 30s, 40s. Alongside this, there's a significant risk of insulin resistance, and signs of this including acanthosis nigricans and often fatty liver and dyslipidemia, as well as frank type 2 diabetes. There's hypertension is a frequent issue, and skeletally there are often problems with the hips, with slipped capital epiphyses occurring and subsequent hip dysplasia. Likewise, scoliosis can be a risk during teenage years. And I've already mentioned the teeth, which need specialist input. Another syndrome you may come across in this category is Seckle syndrome, which I would like to argue should be retired as a term. Helmut Seckle, in his work, which was entitled Bird-Headed Dwarfs, was actually describing a nanocephalic dwarfism, in other words, microcephalic dwarfism. And as you can see from these pictures here, this is very similar to what I've been describing as a microcephalic dwarfism. And I would use Seckle syndrome to describe any form of primordial dwarfism. Generally now it's been applied more to those where head circumference is more affected than height, and there's a more marked intellectual disability, along with a prominent nose and potentially a sloping forehead. And this explains why it's sometimes described as part of the primary microcephaly spectrum, where disorders with individuals with just small heads and a normal sized body extend with the same genes into those with a very small head and short stature. And these are caused by genes involved in centrosome functions such as SET152, CDK5-RAP2, ASPEM, and SEMJ. So switching now to another syndrome, Meyer-Gorlin syndrome. So this is defined by the triad of microtia, as you can see here, these tiny external ears, short stature and microcephaly, and absent or hyperplastic patella, as you can see in this radiograph here, compared with a control radiograph. This condition may not be apparent unless you actually palpate for patella, and was only discovered in this lady here after a road accident when her knees were x-rayed. Patella can be detected by x-ray from about 6 years onwards when the patella has ossified, but before that time it needs ultrasound examination. Here, growth is variably affected. If it's most severe, it can be a Z-score of up to -10 standard deviations below the mean for both head circumference and height, but it can be within normal range in the more mildly affected individuals. Also, intellect here can be almost entirely normal. This lady here was a college student when I met her and took these photos. In terms of other clinical issues, these, as well as ears and patella, other cartilaginous structures can be affected, and neonatally there can be a laryngomalacia and also cartilage issues in the lower airways, which can cause a severe lobe emphysema, which can be life-threatening. But after that time, these individuals tend to be very healthy. The only endocrinologically relevant issues would be mammary hyperplasia as a secondary sexual feature issue in adulthood, or at puberty and adulthood in females. Right, switching now from replication initiation genes to genes which are involved in actually ongoing replication of DNA. I'm turning to polymerase epsilon, POL-E gene, which encodes a gene which causes an image syndrome with intrauterine growth retardation and postnatal growth failure. Here, head circumference often is in excess of height. The M stands for metaphasial dysplasia. A, the most significant medical problem of adrenal hyperplasia, which has sometimes been fatal when unrecognized, and genitourinary abnormalities such as micropenis in males. As well here, an immunodeficiency in this form of image syndrome can occur, which can be so severe as to require bone marrow transplantation, as for this young man now, who's now in his 20s, and in others may not manifest at all. This lady here is actually in her 30s next to an unaffected relative. Here as well, you'll see that there's a facial resemblance which is common across individuals. And in terms of adults, here I'm increasingly convinced that there's a lipodystrophy becoming evident in some of the individuals which are most known to me. As you can see, the substantial loss of subcutaneous fat in the limbs in these two individuals here, as well as clinical evidence of insulin resistance with acanthosis nigricans being present in the armpits and neck of this individual here. Okay, so that's replication genes. Now moving on to DNA repair genes. I'm going to illustrate this with ligase 4 deficiency to contrast this in terms of the clinical complications and management issues here. So in ligase 4 deficiency, we see very tiny individuals with a microcephalic dwarfism. And with time, these manifest individuals start to develop thrombocytopenias and lymphopenias with increased infection susceptibility. And in some cases, this progresses to severe combined immunodeficiency or bone marrow failure. Here, bone marrow transplantation has been frequently used, but requires modified conditioning because these individuals are highly sensitive to radiation, which of course causes double-strand DNA breaks, which ligase 4 is required to fix. As well, because of this impaired DNA repair of double-strand breaks, there's an increased cancer risk, which is actually not the case for many of the other forms of primordial dwarfism. So now moving on to a condition which doesn't as clearly fit into my model of cell number deficiencies as the others, but I suspect will turn out to be the case via cell biology mechanisms yet to be defined, is the MOPD1 or RNU4A tachopathies. So this is the second most common molecular diagnosis we make. Is due to mutations in a very short non-coding RNA of approximately 150 base pairs in length. This short non-coding RNA is part of a complex which encodes the minor spliceosome. So in most eukaryotes, there are two spliceosomes which splice out introns from exons as our coding genes are made. U4-ATAC is part of the minor spliceosome, which recognizes a subset of introns which have a specific sequence at their intron-exon boundaries. Here, mutations at most severe were initially associated with Tabor-Lindai syndrome or MOPD type 1. Here, this is the most profound form of primordial dwarfism with individuals as small as -12, Z-scores of -12. They have short bowed long bones and profound growth retardation, which and often lethal within the first 3 years of life. It's distinguished by severe brain malformations such as pachygyria, lissencephaly, or ageria. Also dry skin and sparse hair. As you can see here, the eyes tend to be quite prominent with full-length lips and a small jaw. And other intracranial abnormalities can include intrahemispheric cysts, cerebral hyperplasia, and absence or partial absence of the corpus callosum. This is the original phenotype associated, but it's actually turned out to be a spectrum disorder with less severe, more viable syndromic forms such as Lowry-Wood syndrome, which is an epiphyseal dysplasia with nystagmus, or Roifman syndrome, where there's a spondoepiphyseal dysplasia, which is also associated with retinal dystrophy, with a cone rod dystrophy, and an immune deficiency, as well as these defined syndromes where individuals which don't easily fit within one of each of these three categories. Key things which help you recognize these are things such as an immunodeficiency, such as with pulmonary infections or hypergammaglobulinemia and impaired antibody responses. Endocrinologically, I guess these are not so high on your radar from the point of view of endocrinological complications, but diabetes insipidus has been reported in a few cases, and in one case, hypergonadotrophic hypergonadism. So that gives you an overview both of diagnosis and specific complications and thus management issues for different forms of primordial dwarfism. I'm finally going to come back to giving you a sort of last mechanistic insight and somewhat changing the model of this reduced cell proliferation and reduced cell number and how that occurs. And this comes from the from thinking about other conditions of growth and the role of epigenetic regulators that have been identified, firstly for the opposite syndromes of macrocephalic overgrowth syndromes. So individuals with increased head circumferences and increased height. And these include genes such as NSD1, the cause of Soto syndrome, and SETD2, which are involved in methylating lysine 36 side chain of histone 3, which forms part of the nucleosomes around which your DNA wraps. As well as these, there is also the epigenetic regulator DNMT3A, which is a DNA methyltransferase, which actually modifies directly the cytosines of the DNA itself. And these are associated with a macrocephalic overgrowth syndrome of Tatten-Brown-Raman syndrome. And here was a surprise discovery for us in that we actually found opposite mutations causing microcephalic dwarfism also in this gene. And these were clustered in a particular region of a gene called the PWWP domain. And these cause the opposite growth disorder to Tatten-Brown-Raman syndrome and are due to gain of function of DNMT3A causing DNA methylation at regions it doesn't normally methylate. And this has led us to think about cell number generation and determining microcephalic dwarfism and disorders in a different way, and thinking of it more like rather than an overall slowing of cell number, that this may actually be due to altered cell fate dynamics, changing cell number with time, for instance, influencing how quickly stem cells differentiate into differentiated cells and their output, and whether the rate of differentiation alters size in terms of both human and organism size. And there remains an interesting puzzle for us to solve here, in that many of the genes which cause overgrowth, which are epigenetic regulators, which are listed here, also in microcephalic dwarfism, also gain of function mutations cause the opposite phenotype, suggesting that these are active regulators rather than broken parts of the machine which regulate growth and may be relevant both physiologically in populations and also in evolutionary contexts. So that, I'd like to leave you with a summary of the key points I'd hope you take away from this talk clinically. Firstly, that primordial dwarfism is a diagnosis to consider when you're presented with somebody with an extreme growth failure, that the diagnosis can be made by measuring head circumference, and that if you have a head circumference and a height of over in excess of -3 standard deviations below the population mean, you should be considering both microarray to exclude microdeletions, an NGS panel for microcephalic dwarfism genes, or more typically now trio whole exome or whole genome sequencing for a panel of genes such as the Genomics England R88 panel to diagnose these conditions. This will get you a molecular diagnosis in about 50% of cases, and this, as I hope I've illustrated to you, significantly informs management of whether cancer surveillance is important, radiation avoidance is important, or whether you should be looking for adrenal complications or lipodystrophy and insulin resistance or neurovascular issues. And overall, the etiology of these conditions is due to inefficient cell proliferation and or cell fate dynamics. And with that, I'd like to acknowledge many people who have helped me learn a lot about these conditions, particularly Mike Bober and Angie Duca from the US, who have assembled a huge amount of clinical data through the Nemours Primordial Dwarfism registry. Many clinical collaborators who are too numerous to list on this slide from UK, Europe and worldwide, and also patient groups where I've had the privilege of meeting a lot of individuals with these conditions and their families. And finally to my funders, the ERC and MRC, and also to my lab who've done the work I've been describing to you today. Thank you for your attention.
Thank you so much, Andrew. That was fascinating and a wonderful overview of all the different conditions. And I wonder, in our own careers, will we only expect to see a handful each of these conditions from an incidence and prevalence point of view? Have you any idea of how many there are in the world, each of these individuals?
So at least 800 families we've ascertained. So I think it's a slightly, the more extreme you get, the rarer these individuals are, whereas the closer you get in terms of Z scores towards the normal range, the more diagnoses, overlapping diagnoses there are potentially, and the more individuals there will be. But I think you're looking at 1 in 100,000 to 1 in a million sort of frequencies for many of these. I probably would have only met in my clinical genetics practice maybe 1 or 2 patients over my career if I hadn't been attending patient meetings and had a special interest in this condition, if that gives you some sense of their frequency.
Yes, so they're, I guess by definition, then ultra rare conditions.
Yes.
Yeah. Okay. Which I guess brings me on to the next question regarding care and coordination of these individuals across the lifespan, because as we've discussed before, and you've alluded to, these are very tiny people when they grow up. So their final adult height will be, as you've shown in your photos, about 1/3 of what would be expected by their genetic parents. So this would bring on a number of problems and challenges in the management of the complications, as you've alluded to, insulin resistance and diabetes, also obviously airway for emergencies. And I was struck by the vascular risk in a lot of these, especially in your MOPD. Was it the
MOPD 2?
Yeah, with the more and more. So how do we approach this as a group?
So I think, so the issues I've seen and have been experienced by individuals, particularly with MOPD 2, is that you end up with these individuals who may be weighing 20 kilos as adults and a height of 1 meter perhaps, being managed in an adult setting where there's inappropriate equipment present, and this tension of our rules base, which is not just UK or Europe but worldwide. But if you're an adult, you're treated in an adult institution. There are issues about blood pressure cuffs, as you say, the most acute situations, which are probably the most concerning. A lack of appropriate equipment, ET tubes for intubation. If you have somebody who's just had a massive intracranial hemorrhage and they're that size, need ventilating and management on an intensive care unit, they're not going to do well in an adult intensive care. So I guess it's difficult to do things except on a case-by-case basis, but being open for paediatricians to manage these adults or co-manage these adults, and having paediatric hospitals which are prepared to accept them in this exceptional situation would be very helpful. I think there's a more broader question about child-adult transition, which I think we all find generally challenging for chronic patients, and that's another issue as well. For instance, taking from child to adult endocrinological issues is a more broad issue, which I think you will all face in your practice. But yeah, I think it's mostly the very acute issues I would be most concerned about. which will be the case for things like MOPD2 or individuals with, significant airways issues.
Yeah, very concerning. I think Charlotte has a question, and there's a question also in the chat. So maybe Charlotte, would you like to go first?
It was more like a comment to. Can you hear me?
Yes, I can hear you.
Yeah. It was more a comment to the care of the adults who are very small or not so tall. I have one patient with a completely other disease with progeria, and he says that it's difficult to be an adult in pediatric care, but it is also difficult to be so small and be in adult care, because, as you said, the blood pressure cuffs are not the right size, and when it comes, we have to go to the almost neonatal.
I'm here. Sorry, I'm my as you asked your question, my computer crashed.
Okay. Yeah, it was just a comment to the care of the adults who are very small, and that they. I have one patient who is very small with another disease who says that it's difficult to be an adult in the pediatric care, but it is also difficult to be an adult so small in the adult care. And it's difficult for us, as you said, with the blood pressure cuffs that are not fitting. So usually when it comes, we have to go to the pediatric ward and borrow a blood pressure cuff for neonatals almost. So it's so difficult. So
yes, I agree. And that chimes with me with one of the adults I was talking to about being looked after in the pediatric ward, which you felt and not appropriately interacted with.
No.
So I think there are issues both ways.
It's very difficult. Yeah.
I guess at least having a hospital which has both adult and pediatric care is probably the key thing.
Yeah. And also I was wondering, which specialist do you think is the most typical who would follow these patients as adults, because maybe not endocrinologist?
Yeah, I think it depends on the issues in terms of their most pressing medical issues as to who will follow them. So, for instance, for image, I would say endocrinology would be a very appropriate specialty with the adrenal issues and potentially lipodystrophic issues and insulin resistance. Whereas, for instance, for MOPD2 with the neurovascular issues, you'd want a neurosurgeon involved and neuro screening. You could make an argument that will have a role. I think generally we tend to signpost to the relevant specialties for the management issues required. I think it depends. I mean, for instance, ligase 4, for instance, because of bone marrow issues. I think it comes down to the diagnosis and the management issues.
Andrew, may I ask, Andrew, do you run a genetics service, clinical genetics service, where you sit down and go through all of the risk factors and signpost these patients, or are you following them over the years?
So my clinical, my research load is my major activity. So I don't have the bandwidth to have a specialty clinic of my own. I'll respond to queries and try and advise and give input. There are, for MOPD2 and MOPD1 now, good gene reviews. Well, in my opinion, good, because I was involved in writing them, but I would hope would be useful management advice for the most common conditions. But yeah, unfortunately, we don't have even in the UK, we don't have a specialist clinic for this group of conditions. Sorry, not to be more useful there. one other thing which I realised I didn't mention with management was early on in terms of growth and feeding. I mentioned that overfeeding, it tends to be a significant issue for these individuals. So for instance, with MOPD 2, you'd only expect a growth rate of 2 grams per day, which is much less than you'd expect for a average stature child if you're feeding them. And the consequence of that is quite a lot of morbidity in terms of reflux and regurgitation, also with tendency to individuals to end up with peg feeding for supplementation. They can end up quite obese to their size with issues because of over-enthusiastic nutritional supplementation. So that's something to be aware of more generally, which I probably, should have emphasized more.
I'd highly agree with that. And I think in Silver-Russell syndrome, for example, there's a move towards discouragement of the overfeeding and just ensuring adequate nutrition for, you know, within their own proportions with their linear growth, and not to be striving to get them onto normal centile charts when they're small.
And I think, I think you know, the issue is that most failure to thrive children, feeding works.
Yeah.
It's recognizing the individuals who it won't. And again, I guess this comes back to measuring head circumferences is at least, I mean, for Russell Silver, it's not going to help you, but for this group of conditions, that will tell you that your ability to compensate nutritionally is going to be limited.
Okay, that's really good. There is a question in the chat. Thank you for your fascinating talk. Are there any clinical studies for treatment of the stature and insulin resistance issues? So I wonder, does that person mean clinical trials that you are aware of, or any particular treatments?
I mean, I guess this is an issue, as you will come across with other ultra rare conditions, is that there aren't ongoing clinical trials. In terms of insulin resistance and lipodystrophy, my colleague here in Edinburgh, Rob Semple, is somebody I would go to for advice on management of lipodystrophies, and I'm sure he'd be very happy to interact with people in that respect. I don't know of any ongoing trials for treatment for that beyond metformin and restricting calorie intake as the main two elements of management. That probably reflects also my limited knowledge in that area, rather than...
You can't do it all. It's hard to do it all. Yeah, that's certainly a challenge we have in a patient with type 2 diabetes with POL1, who is 40-something years old. We were asked to see, who weighs less than 20 kilos. So even dosing for metformin in that would be very unusual for us in a child of that size, let alone an adult. So we're in discussion and thought about that, whether we just do a microdose or something and try, because the other issue is the monitoring side and how much compliance you're going to get with someone who's already very medicalized. It's very difficult. Another question would be about body composition. Do they tend to suffer from osteopenia or osteoporosis? Do you know?
So the fresh in my mind is the DNMT3A individuals with this gain of function mutations who have a very profound osteoporosis. I need to go back more systematically and look through the literature on the others. It's certainly a possibility. It's not something I've systematically asked. With DNMT3A, they end up with minimal trauma fractures. I'm just trying to think the others. It's not something I'm so aware of, but it wouldn't terribly surprise me if there were osteoporosis in some of these other conditions.
Yeah. I guess one of the things is getting the appropriate DEXA, because if they're very, very small, they have to be corrected. for height.
Yeah.
And we had that challenge with this girl I was talking, this woman who's 47 now, where we actually did put her in a pediatric DEXA and got the software to adjust for height, but didn't tell the scan the age. I think it was the only way around it, because the computer wouldn't handle that, but the stature corrected worked, and it was really reassuring. Yeah.
I think the other issue is with blood pressure, as well as what's an appropriate adjustment for blood pressure. And I think body size is probably a better proxy than a chronological age, as for similar reasons as DEXA. The evidence base of that is not.
Well, there's probably no data, is there, for centiles of blood pressures, because in pediatrics we have blood pressure centiles. Do that in these conditions would be very, very challenging without a huge number of patients. I don't see any other questions in the chat. Charlotte, do you have any other questions? We've got 8 minutes technically, so we can wrap up.
Yes, I would like to ask the ones that have a normal cognitive function, how well is society's health service working for them? Are they able to work, or what do they do as adults?
So I mean, again, I didn't really touch on levels of intellectual functioning in a lot of detail. I mean, the lady I pointed to who's at college, I'm sorry, I can't remember her height off the top of my head, but she's probably relatively tall, probably a Z score of -5 or something like that, rather than really, really small. The very small individuals, do they have, is their intellect in normal range or at borderline? We haven't done a lot of formal psychometry. It's also difficult to disentangle how people interact, both families and general public interact when you're the size of a small child. So I think they're social, but also there may be some cognitive issues as well, even if they're very mild. Generally, there's a head to body scaling, and again, I'm going to use mammals as a broader observation here. There is a scaling thing, so you don't need as big a brain if you've got a smaller body for the same cognitive function. So I think there is a compensation when both are equally reduced, but I think even then there is probably some reduction in cognitive function for some of them.
Also difficult to find a work, maybe a computer or something. I
mean, the lady I showed at the start, she lovely conversations with her. She works, she volunteers in an elderly daycare facility. She helps, but she isn't employed.
I guess it starts early on, doesn't it, with the school experience and how they're and their desks adjusted, whether they can carry bags, even just practical things. I know a patient of mine with Paul, the child was transitioning to secondary level, and the secondary school went out of their way to make it an easy transition by giving her a locker that was at ground level, giving her 2 sets of books, and then adjusting her desk before she started school. So it's things like that can make a huge difference to their experience. They're feeling that they're wanted there and included. Otherwise, I imagine that it's very difficult to have a normal educational experience if you're being constantly excluded from things because you're just small.
Yes.
Quite challenging. Even things like public transport, I imagine, are a challenge, like getting up the steps of buses and things, or onto buses.
I mean, I think what I would say about this group of individuals is they're very engaging and engaged, and often I think they overcome quite a lot of barriers because of their engaging personalities. I mean, some of the individuals with MOPD2 have been very athletic as well. There's advantages of being small. You can do things that as heavier people struggle with. But yes, I think there are these practical barriers as well, which would be, you know, also the barriers for these individuals in terms of getting clothing when they're small, for instance. And some of the patient support groups have been, I think, particularly helpful with that, or for instance, identifying dentists who will work with very small teeth, or, you know, finding suppliers of dolls' clothes to find things which fit appropriately when they're very small.
Interesting. Well, on that note, I think we will wrap up, and thank you so much, Andrew Jackson, for your time and wonderful talk. I think it was really a lovely overview of the very rare patients that we see from time to time, or at least things we should be thinking of when we are looking at the diagnostic pathway. So really appreciate your time and expertise in this area. And thanks to all the participants for joining us today.
Well, thank you very much for the invitation. Lovely to see you again, Susan, and yeah, and to speak to you all. Bye for now.
Thanks, Andrew. Goodbye,
Thank
everybody.
you.
Bye-bye. Thanks, Connie.